Might
the
get another
mosaic.
shot before it
Thank you.
comes in. I killed it backing out there, I think.
Whoa. That thing is like half a meter wide.
Yeah.
Try a different angle this time maybe before the dust gets here.
Yeah, that'll help with the model for sure.
Wow.
The hardest part about something like this is the lighting.
Yeah, we don't really have look up lights. And I keep meaning to adjust that one that's a bright spot there. And
Yeah.
we even had a note in the log to do it, and I didn't do it.
What's the sponge above the bottommost heterocone? Is that one of the ones we collected last dive?
So yeah, we think that's a type of mycale.
Okay.
What's different about this sponge now?
MYCALE.
You guys notice?
Oh,
Oh no, it's still there.
dripping.
Sorry, I thought
Yeah.
I'm seeing things. Never mind.
I'm guessing when we do
Andrew
another
says.
mosaic, we want the lasers off, right?
Yeah.
For next time.
You got it, Mike?
Yeah.
Thank you.
Lasers off.
It's a bit dusty.
So a lot of people would like to know kind of the age of some of these sponges, and the truth is that we don't know exactly, but some of these very large individuals we think could be a couple of 100 years old.
Oh, wow.
It is difficult because you can measure growth rates on certain species, especially ones that kind of occur a bit shallower or that you can keep in the lab. They use sponges that have grown on shipwrecks or cables that have been laid at a known date to work out what sort of maximum ages of sponges. But they don't have handy tree rings or growth rings like a coral or the otolith of a fish.
Sorry,
Do the
kids. I've
spicules become more
killed the viz
complex?
there pretty much.
Well, so
That's
the
okay.
spicules get moved around the skeleton of the sponge by specialized cells. So yeah, they can like move the spicules throughout the body of the sponge. So it's not like it gets laid down and it's permanent. And also just the nature of the glass spicules, I don't think we have a good way of kind of aging those. So you have to use things like growth curves or even taking photographic measurements of species so you can estimate changes in volume, because that's the other tricky part too, is you can measure linear growth, but then you also, ideally you want to quantify as those heterocone get bigger, for instance, you're growing all those extra kind of projections on the outside too. Yeah, so some of the 3D measurements that we can get now, that could be useful as well, especially if visiting a sponge over repeated years.
Down.
So one thing we do know is the smaller sponges grow a lot faster than the big ones. So once they get older, their growth rates slow down. So yeah, some of the little barrel-shaped glass sponges can grow a couple of centimeters a year, which is pretty quick.
So we saw the green lunar sponge and it has sort of a softball size or a baseball size, sort of more of a round morphology. How did those grow? Did they just get to be bigger and bigger balls?
I have not read anything on the growth of those. They're Latranculia. They also occur in shallow waters too. So I would guess there's been more studies on the shallow species. Yeah, I think they probably just increase in volume. So there's an interesting phenomenon that they've found on sponges on coral reefs. So these are demo sponges, not the glass sponges, but they a group of researchers measured sponge growth compared with how much the sponges were feeding, and found that if the sponges were using all of that food that they were taking in out of the water column for growth, they'd be sort of enormous, whereas the size of the overall sponge stayed relatively the same. So they were kind of a bit confused about what on earth was going on with why were the sponges feeding so much if they weren't using it for kind of growth. And then they found that they were basically recycling all of their internal cells. So sponges have these specialized cells that they waft. They have little cilia, like little tails, and that's what creates the current throughout the sponge. And they were just basically shedding cells out into the water column, which were in turn again providing food for other invertebrates on the coral reef. So they were providing this sort of link between tiny bacteria and organic matter in the water column, and then through this kind of recycling process they were doing of their own tissues, but then that was being captured by other inverts on the reef. So they called it the sponge loop. So yeah, the sponge size is not necessarily just related to kind of how much they're feeding on. They seem to optimize for the conditions that they're in. So I've seen a sponge in a tank in a lab where one day I'll see it and it looks like a baseball, and then kind of a few weeks later it suddenly has all these, it's changed shape because of the particular currents that it's found itself in, and it's adapted and it's kind of morphed its tissues into a shape to take advantage of the new conditions and food levels and current. So yeah, they're pretty cool.
Opportunistic animals. How cool.
Yeah.
And they are animals.
They are. They're
Bring
very
your head
plant-like,
just
but
to the
they
north
are
just a
animals.
bit for us, Mike.
Copy.
Yep, they filter feed, they produce larvae. So a lot of them are hermaphrodites. So during different stages, they'll produce kind of male and female gametes. Some of them will then brood the larvae in their tissues. There's a really cool work, which was on Asbesta We might see some of them on this dive, the pipe cleaner sponges. So when that sponge releases its larvae, the larvae basically tear through the tissues of the sponge, and the poor adult sponge loses most of its body tissue during that process.
Wow, does it help the larvae?
Well,
It
the
must,
larvae go,
right?
the larvae, I think it's just, yeah, the way that they release the larvae. Well, then the adults, then they, so it's Jackson 2, I think, did this study, and he kept these Asbesta alive in tanks, and then found that the final phase of the adult's life was they turn into what looks like cotton balls on a stick, and they're basically little packets of the adult tissue, which then will then get kind of released out into the water, and those little packets it's a type of asexual reproduction. So then the adult lives on through these little packets that then go and, well, if all goes well, grow new sponges. So asexual
So little
asexual
clones.
reproduction. Little clones.
Nice.
Yeah.
Wow, sponges are so fascinating.
You wouldn't think so, just looking at them, but yes. I'm pretty dominant here as well in these kind of environments.
Erica has asked for a zoom. Is it on the sponge or the coral, Erica?
Go ahead and
I think
you can
maybe.
zoom in there if you want.
I think she wanted a rockfish
Oh,
zoom. But
oh,
I
on the
don't see
rockfish.
it.
Oh, the
rockfish.
rockfish. Yeah, I was like, what's RF?
There's one of those green Luna sponges up.
Yeah. What was that? Latranculia?
Latranculia?
Latranculia.
Yep.
You're gonna have to type.
Or
Which
is that
fish?
a yellow mycale? Just a small one.
Oh, that little one. It could also, I've been tricked before sometimes, like the shells of like brachiopods or clams look a bit yellow. Or are you
It was
talking
a
about the one
rockfish,
that's
but it's not
underneath
there
the
anymore.
Swiftia?
I was talking about the one that's above and to the left of it, but it does look like
Yeah.
a brachiopod now that you
Oh,
say
yeah.
that.
No, being tricked.
Oh, there's the
before.
rockfish.
Yeah,
Is that the one she? Yeah.
I've been seeing a lot of this kind. I wonder what it is.
I think it's the dark blotch. So we shall see.
Oh, she says the one below without the stripes. Is there one below?
Not seeing it.
It probably means like
Looks like there's a nudie there. Sea slug,
Yeah.
maybe, or is that an
Tritonia,
anemone?
maybe.
that one.
Tritonia.
That one?
They all have stripes.
Yeah, that does also have stripes. I don't know, maybe it's gone.
Oh, I've just lost the science chat.
And I keep moving to keep ahead of our dust clouds.
Are we? What waypoint are we near, please?
Yeah, we are still between 3.02 and 3.03.
Great, thank you. At 3.03, we're gonna make a detour up to explore some seeps.
3.04, I believe.
Oh, is it? Okay, yep.
We could go from 3
Great,
actually
thank you.
if you want.
Yeah, it's, yeah, zoom
I
out.
think they were
Oh, yeah,
renumbered,
it's not
so
bad
I'm
from-
probably.
They
You could
weren't
do it from
renumbered, Jenny, just so you know.
either.
We did not renumber them.
Oh, 4 might be closer. I don't-
3
it's
or
Four,
four
four is
fine.
closer, but either way we can get to it. So however you want, if you want to go up this ridge or if you want to go in this like crevice here.
We'll decide when we get there.
Sure.
Jenny, did you hear that? If we go up from point 3, it takes us along a ridge. If we go north from point 4, it takes us kind of through a valley.
If you have the quad
Ooh,
up, I
I'm-
can point it out.
Abby, what are you thinking? I'm tempted to go up a ridge instead of a valley.
Yep.
But
Yep, look at the draw, but steep sounds good.
Sure, we can try this prom. Just kind of go up it that way.
Use another bump. Ready,
Roger.
please.
Wow.
nice space. Look
20.
at that.
Yeah.
beautiful.
20 towards you?
Yep.
Let's see, you're facing 315.
Yeah, I'm looking west. Argus is looking 315.
I want to eventually get not that direction. Bridge nav.
Which way you want to go?
20 meters, 315. I think north and then east. So I'll show you. Let me.
We can go. I can go north.
Here,
Come
I'll
up
show you.
out of the muck here.
So these contours, we want to eventually go here. So
Roger.
we're kind of following that around, but
Chase it around. Yeah.
yeah. So let's see, where are we going?
But we're definitely going tourist speed. This is beautiful.
Yeah, there's so many of these fully
There's
dead
a
for
line
a
down there.
Yep.
and other glass sponges that kind of make up parts of the wall.
A couple of them actually.
And then those big heterocones interspersed.
Yeah.
Is that cable?
Is that line?
Yeah, there's our line.
Yeah,
it is.
that looks like line on the wall.
Stay away.
Yeah.
Just what we were looking out for.
Yeah, multiple.
I
There's
think
one on the
that's
right.
two.
Yeah,
Yep.
that's alarming.
Wish there was some way to remove it.
There is. You get it tangled up in the ROV. Bring the whole mess to deck.
Yeah, let's not do that. Let's. How about a good way to remove it? That would not be my first choice.
All right, do you want me to cancel that and go north?
No, I'm all right.
Okay.
Can we
Fault
look
line
at that
is
big boy
down
down
over.
below? It's got to be a meter, close to
Yeah,
a meter
copy. Or
across.
I can look from here, but we're We're moving the ship, also waiting for Argus to catch up.
Okay.
I kind of zoomed out of the poor visibility area to some fresh water.
Mm.
It's really mucky down below for some reason.
Yeah, we haven't even been close to the wall.
I think there was a major avalanche. Like, truly.
Seems like it.
I think it's all those fish.
Yeah.
It's probably the
Yeah, we're
fish.
freaking out the fish. That's it.
I can have a look. It'll just be a little dusty.
Yeah, at least a ½ meter wide.
Yeah.
Put the lasers on him.
So there's a study, it's quite old now, but they looked at the role of sponges in this was shallow sponges again after a hurricane when there was a lot of dead material and sediment in the water column. They found that sponges, although they initially stopped pumping after the event, once they started, they significantly contributed to clearing the water of
Hit the porch light,
all the
Mike.
bacteria and organic matter in the aftermath of the hurricane.
Could you cut the lasers, please?
Yeah, Roger.
Thank you. Beautiful.
Mm.
I love seeing it illuminated from outside
Yeah.
and coming through.
Yeah, I was just about to
Beautiful.
say, great lighting.
We are the new resident of this sponge.
Yeah, that's the camera in the sponge.
Yeah.
Yeah, careful
the
now.
shrimpy lobster.
Can zoom in there, see what it looks like.
Goes on forever.
Down the tunnel.
Yeah, seriously.
It's a small
That's
world.
cool.
It's like a
after all.
weird Twilight thing.
Whoa.
I feel like we're going into an ear canal.
That's gross.
Honeycomb, heterochrome.
Now I cannot not see that.
Have you ever been this close to a sponge?
Earwax.
So
Wow.
close and
Thank
yet
you,
so
guys.
far.
Wow, up close and personal with the sponges of Grace Canyon.
It's not the best reality TV show, but I'll watch it.
I'd watch it.
I got that one, Nicole.
It's
Huh?
core
What
sample.
would you give it?
Core sample intervention.
Really? Oh, I'm so harsh. I gave it a 2.
2. Nobody's even going to look at it.
Really?
Abby, what would you have given that as a rating for highlight video on a 1
At-
to
on a-
5 scale?
On a 1 to 5 scale.
Oh, it's a 5.
See?
Okay,
Man.
see, I'm telling you, the Papa cruise, it forever ruined me because there it was like sponges larger than Hercules, and we were like, that's a 5. I'll be more generous.
Yeah, you have to look at the, we've been on the seafloor for about 2 hours. It's been a lot of silt, but that was a 4 or 5.
Some contextual.
Oh, wow, that's a big,
big
dead
dead one?
heterocone, yeah.
Oh, wow. Yeah, that's he's
Up north?
toppled over.
Sure.
Bridge now. 20 meters north. Thank you.
Can't turn there, don't quite have enough tether to.
Nope.
Without dusting it.
And another avalanche.
Yeah, I think just the slopes are so precarious here that,
Yeah.
I mean, I don't really think that was from way above you.
Well, if I fight the tether, it puts in a lot of the laterals, and it just, once it hits a cliff, it's like a
Right.
pressure washer on. They used to mine
Our
back
hanging
in the
mud
day.
walls.
Strip mining the subsea wall there.
Dead ones up here. Oh, maybe one live one, one dead one.
I can wait for Argus there a bit.
Wow, that is a huge dead Berea.
Mm.
Oh my gosh. These are cool.
Do a DVL reset there. Wow, there's a whole lot of them.
Yeah, so many right
Yeah.
here.
Yeah.
On the last dive, we were seeing so many different kinds of sponges, and here there are different kinds as well, but it seems like the header cones are so dominant.
Yeah.
Is that a factor of depth or something else?
Well, two of the things that would be really, really helpful to know is sedimentation rates, because that's something that, I mean, depth, you do have zonation of sponges with depth, but often that's just, it's because you have the various different environmental parameters changing across depths. And the things that we know are really important for sponges are current and sedimentation. Those are two major ones. So I would guess that it's something to do with differences in the currents that you had at those two different sites. I mean, we know that Gray's Canyon, you know, has high levels of methane seep, so that's been kind of put out there in the literature that it could be associated to the, you know, being able to, maybe some of these sponges are able to feed on the methanotrophic bacteria, but I mean, that's just a hypothesis.
Yeah,
So
because
it
you
could be
keep getting
something
up
as well
ahead.
to do with that.
Look at that one
Whoa,
off to the
huge
one
right.
coming
That's
up.
the largest one I've
0.045 or something.
seen.
That one is massive.
Once we get up over there, once I'm closer to over you, I'll back off, and then I'll...
If it wasn't so so turbid, we would probably be able to see some really cool shots of these sponges next to Herc and
Could
Argus.
you please pan up before we lose Viz? I'm sorry to interrupt you, Leela. That one is.
This is even more in the distance off to the right as well.
Yeah.
Oh my god,
Yeah.
there's a huge one.
Wow.
We're trying to reposition Argus, so I don't have to.
Okay, thank you.
Excuse me.
Sorry, I'm not as operationally aware as if I was on the ship.
No, we're trying to reposition Argus so we don't have to adjust the vehicle heading and blow out the cliff there.
Yeah, I'm
Should
gonna,
improve.
we're still finishing that move north, and then I'll step off a bit.
Is this a good one for a pirouette, perhaps? Or maybe the next cluster?
Have a look, yeah, the next.
To the right.
Yeah.
Yeah. I like that idea too.
Bridge nav.
Can zoom in there if you want, Marley.
Roger.
That's good.
10 meters, 045. I think that ought to help.
Bring your head to 315, would you, please?
Yeah, I'll keep you inside.
So was a pirouette possible here, ROV?
Yeah, once we get Argus
I think we're
up
gonna
behind
try.
us, We need
Yeah,
to
for
get
the
it out
next
there
little
anyways.
cluster.
What was that, Jenny?
I think we're gonna try for the next little cluster.
Oh, the one
if
behind in the back?
I can zoom
Yeah,
out.
if the viz holds.
Are we zoomed out there? Thank you.
Wow. Is that one of them? I thought that was multiple, but I think that's just one.
One at the top.
I think it might be one.
Yeah.
Wow.
So one of the questions we had after NA072 was how extensive, how far these huge glass sponges extended. So it's pretty good, pretty interesting to know that they kind of continue down the canyon as well, not just on the top.
And another 10 meters north,
Yes, please.
bridge nav. Ten meters north. Bridge nav. Add one more step, 10 meters north.
Which one were we gonna do then?
I think they'd been
Maybe the big
eyeing the
one
one
on
in
the
front of
top.
us. Is that what you guys were talking about?
I think so, yes.
Roger.
I don't think this is gonna be clear enough. Do you think it will clear up more?
If I get closer, it'll be a little better. But yeah, it's not gonna be crystal clear. I think the current's kind of going with us, you know, so.
Mm.
So everything we've disturbed where we've been is kind of chasing us.
Well, we
So
got some nice shots of it anyway.
I can come around it here while we're looking at it. Like we might be a bit overexposed there, or is it just me?
Yeah, I'm adjusting kind of constantly. It's hard with the sediment in the water, but
Yeah, right.
just let me know what you're looking at it on a much bigger screen, so if you think it's overexposed, just holler. Does that look okay to you guys?
Yeah, that's better, thanks. So,
That looks good.
You might need to bump west.
Roger.
Bridge nav.
It's gonna turn to the other way here soon, so.
10 meters west. I think that'll get us closer, then we'll move north and then east around this little.
Probably should have had you wait till they come around here before we move it, but. It's starting to get tight there.
Yeah.
I'm having to give more command to. Which way did you go? To the west?
I went to the west to come.
Oh, yeah,
towards it.
perfect. Yeah, thank you. I'll wait.
Argus can flip if you want.
Yeah, if I give it any more jam than that, it's gonna blow it out.
Yeah, yeah. Views from the bottom up.
Yeah, there's no light that way.
Yeah.
Jenny and team, we've been flying without lasers, but if you want them back on, let us know.
Yeah, thank you. Now that we have taken some nice shots of this, it would be great to throw the lasers back on, please.
Copy. We're gonna come around it once more. We're still positioning Argus to.
Should we try to rapid fire a little this time?
Yeah, after this move, we should have enough to come around it without excessive thrust, and that's cleared up a bit. So.
On the heterocone that's right below it, it has this fun little knob on the
Oh,
top
yeah.
that sticks up.
It's got
Looks
a
like a little
boot.
wave. Mm-hmm.
Okay, here we go. Let's see if I can sneak to the right a bit more and then pirouette around again.
So Abby, all of those additional knobs greatly increase the surface area of the outside of the sponge. Is that why would the sponge do that?
By design, I think, Jenny. So yeah, sponges are all filter feeders. So they have this, the name porifera means pore bearing. So basically they're just full of these tiny canals, and that's the way they feed, is they have these specialized cells with little tails that beat and create a current through the sponge and will trap particles. So the greater kind of their surface area on the inside, the more surface area they have for feeding and catching and trapping food particles. So by having those little finger-like projections on the outside, they're increasing that surface area. So yeah,
More mouths, more food.
Yep. Yep.
Not to mention very interesting to look at.
Sablefish.
Okay, not doing a head plant into the substrate.
Visibility is much better now.
It's beautiful.
Yeah, we have Argus right behind us, so we don't have to fight to bring our head around. Nobody yanking our leash at the moment.
So pilots, when Herc got tangled yesterday, was Argus caught up in that line as well?
The tether between Argus and Herc was what we
So
got tangled up. Well, there's it's a 30-meter tether, and there's 2 floats and a weight. So the floats and the weight are evenly spaced, roughly. And it got caught on the float closest to Herc. And then when we cut that first line, it slid down the tether right to the back of Herc, which
Like the pot was on the back of Herc.
yeah, it was. The rope was around the tether and then anchoring us, and then there was a float which we had in our left hand. So luckily, when we finally cut the big line, it came off the tether, and we just let go of the float.
Could we maybe go a little bit lower on the next pass around?
Yeah, sure.
Pass east, just to get some of that underside complexity. Thank you.
You want to look to the left with bubble in there?
Oh, I was just about to ask you.
Thank you. I think that's about it. Okay, you can look back in the middle.
Of a flatfish.
And look back to the left for me, please. Thank you. I might, if I back up a little, I might put the light on it better. It's the dust.
Hopefully the high contrast of the white sponge with the dark background will make it easy for the software to stitch these together.
That was the rockfish dusting us there. Maybe a little bit of Herc dust too. What? Okay, that's twice around. No.
Thank you. Could you throw
Are
the
we good?
lasers back on when
Sure.
you're done with the mosaic, please? Thank you.
Lasers are on.
Actually, can I run through gauges real quick?
Yeah. So moving on then.
Yeah, let's
That
keep
sounds
on
good.
exploring.
So I'd like
And,
to move north along here, and then eventually we'll go east.
Hit that dog
right
leg
in this
up there.
crux.
Yeah.
Yeah,
I'm wanting to get there too. Hopefully the current will be different.
Roger. I can start a 20-meter north move now. That way we'll stay in this orientation-ish.
Sure.
Bridge nav. Two 0 meters bearing north.
Another huge dead mamba farrier with some little live bits.
do you think they're doomed, or do you think a sponge like that could recover?
Well, the fact that we've just seen a lot of the farrier dead makes me think that maybe conditions here have
Whoa.
changed from when it was optimal.
Go, Caesar, go.
So I think it might need, oh, look at it go.
Bye, Rathbanaster.
That's why it's great that Herc has a suite of environmental sensors attached. So we'll be able to, once we get enough surveys, we can look at correlations and abundance of the different sponge types with the environmental parameters that we're measuring and try and work out which type of conditions are favoring which sponges.
Coming up.
You're stuck in a corner.
Stuck in a corner. Where'd that corner come from? Snuck up on me.
Yeah, it really did.
It's always tricky because you have environmental influences on species distributions, but also biological factors such as, you know, their reproduction, and
Goodness.
quite often those are things that we really just, we don't know enough about in detail for the species involved to be able to kind of see how much those factor in.
Marley, can you zoom in just a bit to get rid of the push core there?
Yep.
Thank you. I gotta look down a little because the light is killing me.
We do know that the currents up and through these canyons will transport nutrient-rich waters up the canyon, which is probably part of the reason why we're getting such large sponges. The big dead ones.
Wow, that large one must have been there for hundreds of years before it died.
Still providing structure though, even once it's dead.
Can other sponges settle out on an old sponge that's dead like this?
Yeah, they can do, and that's kind of the process that forms sponge reefs. Not seeing it so much here. I think that the conditions where they do that, there's differences in the geology. So the sponge reefs are often found in areas which were scoured by icebergs, and they form along the scour where the icebergs scoured the seabed. They also, I think most of the sponge reefs have been found further in kind of more sheltered environments, further inshore. So yeah, up around Vancouver Island, where actually you do have quite levels of sedimentation, but something about the conditions there has enabled the survival of these sponge reefs, which were kind of thought to be extinct until the 80s. There's also kind of differences in the silica concentrations in the waters as well, which will play a role in that too. Yeah, it's an interesting question where we don't see these big bioherms of sponge living on sponge. Probably a combination of factors.
Yeah, with sponges and corals, I'm often quite concerned about mechanical damage from things that interact with them, whether it's traps or nets or cables or whatever it might be. But it's
ROVs.
interesting, and Liz noted it as well, that
Oh yeah.
they haven't been crushed or mashed or mechanically damaged.
Yeah, I mean, I think they're pretty vulnerable to that. Sponges can regenerate from small amounts of damage. We know that their regeneration rates are a lot faster than their growth rates. Again, I know more about demo sponges than the glass sponges, but in a demo sponge, their cells can actually swap from one cell type to another, kind of like our stem cells do. So if a sponge tissue gets damaged, they can just recruit other cells to that area to kind of regrow that damage to the sponge. If a trawl went through and knocked a sponge over, then that would likely be game over. But maybe the canyon's a bit steep and there hasn't been a lot of trawling at this particular site. I know that the spot prawn fishery is largely with pots, which would be less likely to damage things like these big sponges.
Okay, I'll go a bit north here, about parallel again.
Roger.
Bridge nav. 20 meters north.
Yeah, the fact that we've got a mix of live and dead sponges here too, and that you can still see the structure of the dead ones makes me think it's probably more of a just a natural process. You'd expect to see some live and dead individuals, particularly with this species where the skeletons will stay intact after the sponge is dead. So for most types of sponges, they'll just fall apart and all you'll be left with is kind of spicules in the seabed. So I think that actually might be some dead mycale at the top of this sponge where you can actually start to see some of the exposed skeleton. But that's an example of a sponge that will just kind of fall apart and won't stick around like these big heterocone skeletons.
little radar antenna on top.
It could be my Kaylee Laveni. Oh, Swifty fan, maybe.
Come down 5 place, Mike.
Copy.
Quite steep.
Whoop.
So I need to get on with it, don't I?
Yeah, looks like.
So starfish having a sponge salad, or is he just hanging out?
It could be having a snack. Sponges don't have many predators, but there are some sea stars that will feed on them. Nudibranchs as well. Some nudibranchs even get some protection by absorbing some of the chemicals that are produced by sponges. In the tropics, there's a few fish, angelfish, that will feed on sponge.
I don't know if that one's why that particular one is all brown, because starfish has been hanging out there. It's pretty fat starfish. Sea star, sorry.
Yeah, I did a study where I was looking at nudibranch predation on the shallow water sponges, and you'd look out for little feeding scars. So the nudibranchs have different kind of feeding gear, and you could often tell, like, some of them would produce this little stamp shape where they had attached themselves to the sponge and then spat out sort of like digestive enzymes and then slurped it all up like a fly. Other ones, they will like scrape off the surface tissues of the sponge. So I'm not sure what a sea star feeding scar would look like, but those were the clues that I would look for to tell if something was actually feeding on it or whether it was just using it as a perch. The fact that it's dead, though, could be something.
See if we can make it around here. start to get up the ridge.
Liz is commenting, well, looks like we're seeing some small Africalystees vastus, which is a different type of glass sponge as well, present in the mix. Dead barrier. But dark blotched. Think another Michaeli mapping us there.
The one at the bottom.
Mm.
Okay. Mhm.
The dead Hetrico in there. No, I'd say we're definitely seeing more dead barrier than live. All the little opioid arms sticking out. Yeah. Yep, we have not seen anything alive like that.
Really big sablefish.
Another 10 meters north if you want to get in.